Well, good morning, everyone. A few housekeeping items. If you're an Emory University healthcare employee and you would like to receive CME for attending today's talk, the access code is 701966 and can be found at the chat feature in the bottom of your screen for virtual attendees. And if you have any issues, please send Katiatu Fofana an email or drop a note in the chat feature. It gives me an immense pleasure and excitement to introduce to you today a remarkable person, a remarkable researcher, a remarkable innovator. And if you're in the cancer field, your way of diagnosing, of classifying, and our way of diagnosing, classifying, understanding cancer, and most likely treating cancer over the next 10 years will clearly be affected by Anant Madabhushi's work. If you're an Emory employee and you have not heard yet of Anant or his work, your employment probably should be revoked, as you probably live in an alternative universe, as Anant has already, impacted our research processes here, especially in cancer. I'm going to be very brief and respectful to Anant because he asked me to be very brief as well, and in respecting to the audience too. Anant is the leader of the Emory Empathetic AI for Health Institute, or AI Health. He's a global leader in developing AI to improve outcomes for individuals with cancer. He's a fellow of the American Institute of Medical and Biological Engineering, fellow of the Institute of Electrical Engineers, and a fellow of the National Academy of Inventors. He authored more than 450 peer-reviewed publications. He holds more than 100, or is to hold more than 100 patents, which gives you an idea of the scope of work that he's doing globally. And so I think no doubt that his work will clearly impact our cancer management. And the title of his talk today is very correctly inspired by the Sid Mukherjee's very best seller and very well-known book describing the inception of understanding of cancer back since the 1960s until today. Please join me to welcome very warmly Dr. Madabhushi for his first Winship Grand Round.
Thank you, Nabil, for that very, very kind introduction. Extremely flattering. And I want to assure everybody that it's going to be all downhill from here on. So just he set the stage and I'm going to really bring it down. No, but really a great pleasure to be here. It's been amazing to think that it's been a year and a half since our group moved to Emory and to Atlanta, and thrilled to be working with a number of folks in the room and possibly virtually. And the goal here was really to introduce the portfolio of our work around artificial intelligence in different cancers. And really my intent here is to try to engender even more collaborations and work even more closely with the Winship Cancer Center. So, yeah, so just want to get the conflict of interest disclosure out of the way. Just want to maybe mention that Picture Health is a company that I co-founded and have an equity stake in. Some of the technologies that I'll be touching on today have been licensed to Picture Health. I also have a number of sponsored research agreements with pharmaceutical partners, and those are listed out here below. I guess my true conflict of interest disclosure is the fact that I do have a social media account. That's my X handle, if any of you are on X, and any of what I present today is remotely appealing, please feel free to give me a follow. Right, so just to set the stage a little bit, I think that all of you are acutely aware of the issue that we have in cancer, which is an overdiagnosis and overtreatment of a number of cancers. Prostate cancer is a great case in point, where 1 in 7 men in the United States will be diagnosed with prostate cancer, but 1 in 40 will die on account of it. And yet, Unfortunately, today, we still throw the proverbial kitchen sink at a number of these more indolent cancers. With prostate, we still tend to subject a lot of these men to radical prostatectomy, radiation therapy, and sorry. We still subject a lot of these individuals to the toxicity associated with more aggressive treatments, even though a lot of these men may have more indolent disease. And apart from the patient-centric toxicity that this brings, there's also the very real issue of financial toxicity that we have in the United States, which again, I'm sure a number of you are aware of. Something like 42% of our newly diagnosed cancer patients will go bankrupt, many within 1 to 2 years of that initial diagnosis. So the focus of our group has really been on how to leverage routinely acquired data with artificial intelligence. And again, I use the example of prostate here. Over the last decade and a half, there's been a lot more interest in looking at non-invasive imaging like multiparametric MRI. And you can see here in region of adenocarcinoma on a T2-weighted MRI scan. With a multiparametric MRI exam, one can acquire different parameters, different sequences to characterize the tumor. But assuming this individual goes on to get a radical prostatectomy, we now have the ability with whole slide digital scanning to create high resolution digital image representations of those pathology examples, and essentially start to bring AI and machine learning and image analysis algorithms to start to interrogate the morphology of the tumor in a very quantitative, highly specific manner, allowing us to really tease out features of cellular morphology, individual cells, shape of individual cells, and really capture a plethora of features and patterns that span the multiple different scales, from the macro level scale from radiology down to the cellular level scale with histopathology. And of course, as is happening quite routinely for a number of cancers, we're also collecting a lot of omics information, either through proteomics or genomics in the clinical space, and then certainly in the research realm, more single cell and spatial transcriptomics. And so with all of this information across these different scales, it really presents a tremendous opportunity to think about how we can firstly align the knowledge across these different length scales. How can we then extract features and extract patterns across these different length scales? And ultimately, how do we fuse and combine the representation across these different length scales to be able to create improved integrated predictors of diagnosis, prognosis, and treatment response? Today, when we talk about artificial intelligence, I think a lot of us are really being told that there's sort of one AI algorithm. And I want to sort of dispel the myth here. There isn't one AI algorithm, right? A lot of what we hear today is specifically about one category of AI called deep learning, right? So deep learning essentially is an idea built on neural networks, and I'm not going to bore you with the minutiae here. But the idea of neural networks has been around for over 60 years. And what has happened over the last 10 to 15 years is that with advanced computational horsepower, with the ability to essentially map these neural networks in more sophisticated ways, we're able to create very deep networks like the one that you see here that allow for encoding of knowledge and encoding of information. So essentially, the idea here is you've got all these nodes that represent this neural network architecture. You can provide data to the neural network, and the network then is able to encode for information in the data in a largely unsupervised manner. This is actually work that we published about a decade ago in the context of breast cancer. One of the very early examples of the use of deep learning for breast cancer, and in fact in cancer in general, where we essentially provided a series of different pathology images to the neural network. And my student who was working on this at the time, who didn't know anything about breast cancer, knew nothing about pathology, but working with a pathologist was able to get annotations of individual cells on these training images. Those were then provided to the network. The network then learned representations of the individual cancer cells and then was able to identify the location of these cancer cells on new unseen images. So this was fascinating because with very little domain knowledge and simply having annotations of the individual cancer cells, we were able to stand up a network that could now go and find cancer cells on breast pathology images. So this in turn has spurred a great deal of activity in our group. We've managed to make these algorithms far more efficient today. So this is off just standard off-the-shelf hardware. We can now run these algorithms. You can see this on a whole slide image. It's about 40,000 pixels by 40,000 pixels. The ability of these algorithms to go in and find lymphocytes, to find cancer cells on pathology images very rapidly, right? And so essentially what we're able to do now is with off-the-shelf hardware, we're able to digitally phenotype these pathology images in a highly quantitative and highly rigorous manner. Now, the caveat, though, is that a lot of these deep learning algorithms are referred to as black box or opaque algorithms. And the reason is because the representations that we get from these algorithms are very difficult to really fathom and interpret. And that is a very valid concern, particularly when you think about in the clinical space, certainly in the oncology space, where you're being asked as an oncologist to modulate somebody's treatment regimen based off the predictions of an AI algorithm, where you don't really understand what the representations are. What do these features mean that you're bringing to the table? And so there's been a lot of concern around the opacity of these AI algorithms, but beyond the lack of interpretability, there's also real concern that the lack of explainability is driving a lot of issues around reproducibility in the AI space. And there's been a series of articles in Nature and elsewhere that have talked about this sort of reproducibility crisis that we're seeing in science. And a large part of it is being driven by the lack of interpretability with regard to the AI algorithms. Because if we don't understand what the AI is really picking out from the data, then when the AI fails, you don't really have a sense of how to fix the issue. And the typical response is throw more data at the problem, and hopefully more data will fix it. But fundamentally, when you don't understand what the particular representations are, then it leads to sort of this, as is termed here, sloppy use of machine learning. In our group, we've tried to move away from that and really focus on more explainable artificial intelligence and explainable AI approaches. And our intent has really, a large part of our portfolio, as you will see today, is really focused in the cancer realm. And while there's been a lot of interest over the last dozen years in the diagnostic realm, so being able to develop algorithms for identifying disease early, for better diagnosis, all of which is very important. Our group in particular has been very focused on prognosis and in particular, predictive analytics, being able to not just diagnose the presence of the disease, but also be able to predict disease progression, predict disease outcome, stratify risk, and more critically, be able to develop predictive algorithms so we can identify which patients are going to respond to certain treatments so that we can ultimately use these algorithms to really truly realize the promise of precision medicine. But to do that, we need to address the reproducibility crisis. And certainly the approaches that we've taken in our group have been essentially fourfold. One, focus on more explainable AI approaches, and I'll give you examples of what these more explainable AI algorithms look like. Focusing on equity. This has been a major focus for our group, and I'll pepper a few examples in there to talk about how we need to ensure that these algorithms are working across different populations. And then as working with Nabil, with Ram and others, really focusing on trying to validate these algorithms on completed clinical trials, right? And so this is where we've been working both with pharma as well as cooperative oncology groups to be able to first validate in a blinded manner on trials that have been completed. How are we able to predict response, predict outcome? But then ultimately, the proof of the pudding is in prospective validation. And I'm happy to report here that we've started in a very nascent way to be able to start embedding these algorithms into prospective trials. And I'll show some data from an ECOG clinical trial, but also working with Nabil and Mihir and others, thinking about how we can start potentially within Winship, investigator-initiated trials, where from the get-go, we embed these explainable AI algorithms, either as secondary or exploratory endpoints, so we can start to generate prospective data. All right. So the explainability of the AI approaches. So I suggested that there is this issue of opacity and lack of explainability with regard to AI, but it doesn't mean, I'm not suggesting we throw the proverbial baby out with the bathwater, right? There is a great deal of power in these algorithms, but the way that we're using these algorithms is rather than saying we want to make a prediction from a pathology image or a radiology image as to what the outcome is going to be, what the treatment regimen should be, or what the treatment response should be, we're taking a slightly different tack. We're first leveraging the power of these algorithms to do what we're calling digital deep phenotyping. So you take a pathology image, we leverage the power of AI to go in and segment out the different tissue regions on the image, which means essentially segmenting out the tumor, the stroma, the epithelium, the different tissue compartments. From that, we can then extract the individual cells. We can look at lymphocytes, we can look at cancer cells. And having done that, now we can really start to prize out more interpretable features, features that relate to the shape of the individual cells, relate to the number, but also relate to the arrangement. Start to look at spatial statistics. What is the co-arrangement between lymphocytes and cancer cells? And really converting all of these measurements into digital biomarkers that we can then link to outcome and treatment response. And not only can we do this off pathology images, we can also do this off radiology images. Here's a chest CT scan where we can, again, leverage the power of AI to segment out the nodule, segment out the tumor-associated vasculature, segment out the peritumoral regions on these images, and start to pull out a series of features that relate to the heterogeneity of the tumor, but also the heterogeneity outside the tumor in that microenvironment. And then be able to start to look at features relating to the vasculature of the individual nodules and start to look at the twistedness and the convolutedness of the tumor-associated vasculature. And again, convert all of these measurements into quantitative features, and these features can then be related to outcome and treatment response. And over the last half a dozen years, our group has been taking these digital phenotyping approaches, both with radiology and pathology, and also integrating them to look at a number of different cancers, and I'll try to cover some of these. But really looking at it from, like I said, that more predictive lens, being able to predict response to various different types of therapies. And you can see the different classes of therapies there in the middle, and then also the individual combinations way out there in the right panel. I'm not going to belabor every one of these individual combinations, but I just put this out there to show the power of these algorithms in being able to predict response across a whole plethora of different combination therapies. All right, so let's dive into some specific examples. I want to start with some of the work we've been doing in the area of breast cancer, and currently we're partnering with Kevin and Sunil Badwe in pathology. But this is work we've been doing for over a dozen years now, and specifically we started looking at early stage ER-positive breast cancer. And of course, as all of you are aware, one of the big challenges with early stage ER-positive breast cancer is really the question of adjuvant chemotherapy. Which of these patients are going to benefit from adjuvant chemotherapy and which patients can avoid it? And until about 2 decades ago, there really wasn't a good solution until Oncotype DX came along. And then you had a 21 gene expression assay to be able to identify, based off the risk score, as to which women had more aggressive disease and therefore would benefit from adjuvant chemotherapy, and conversely, which women had less aggressive disease and therefore could safely avoid the adjuvant chemotherapy. The problem with this test is that it's tissue destructive. It requires physical shipping of the tissue blocks to Redwood, California, and it's a $4,000 test. And so the focus in our group was, could we do better? I grew up in India, and being an immigrant from India, one of the things that I always think about is the global context. Is a technology going to have an impact in a place like India, where the cost of chemotherapy is about $1,900? How do you justify a test that's going to cost you $4,000 to tell you whether you need chemotherapy or not? About 14 years ago, working with oncologists at the Cancer Institute of New Jersey, we came up with this idea of the image-based risk score. And the main idea of the image-based risk score was to try to come up with predictions akin to Oncotype DX, but to do it of digital pathology images. And so we came up Just like the Oncotype recurrence score, we came up with the image-based risk score, or IBRIS. And the idea was that you take these pathology slides, H&E slides, those could be digitized, and then we could subject that to AI-based analysis, find patterns on these H&E images, and then relate that to prognosis and treatment response and treatment benefit. But one of the things that we wanted to make sure was that as we were building these IBRIS algorithms, that we encoded the features within this with explainability, with interpretability. And so a couple of years ago, my student, Haojia Lee, started to look at this particular problem to predict survival from early stage ER-positive breast cancers. And we started to focus on collagen, right? So if you think about collagen, it's a protein, of course, and it's been implicated in a number of different tumors. And so Haojia started to look at the tumor nest. So she used machine learning to identify the tumor nest, and then also identify the collagen strands on the routine H&E images. So she did a reasonable job, not perfect, but a reasonable job in identifying exactly where the collagen fibers were. And then having identified the collagen fibers, essentially ascribe a vector or orientation with respect to the collagen fiber vis-a-vis the tumor nests. And essentially, what we were then able to do was to calculate the concept of disorder or entropy. So if you recall your high school physics, entropy measures the amount of chaos in a system, right? So if you've got a high degree of disorder with regard to these collagen fibers, right? If they're discordant with respect to each other, entropy is going to be high. On the other hand, if all the fibers are arranged in the same direction, then the disorder is low, right? And therefore the entropy is low, right? And so when we essentially looked at calculating the disorder with regard to these entropy orientations or entropy fibers, we found very interestingly that patients with short-term survival had very coherently organized collagen architecture. The collagen was beautifully preserved. On the other hand, when you looked at patients who tended to have longer-term survival, more favorable prognosis, there was a high degree of disorder. There was a far more chaos with regard to the orientation of the collagen fibers. And initially, this seemed very counterintuitive until you appreciate that there's been a lot of data, a lot of literature suggesting that there's a strong role between metastasis and collagen, suggesting that disruption of the collagen architecture actually impedes metastasis. And on the other hand, better preserved collagen architecture actually promotes metastasis, almost sort of providing that highway for migration of the cells from the primary site to metastatic sites. And so we showed in this paper from about 3 years ago that looking at completed clinical trial, ECOG 2197, as well as patients from the TCGA, that this single feature, just the entropy associated with the collagen architecture, did a remarkable job in being able to predict survival in these patients. We followed this work up with a subsequent publication last year where we went head-to-head with the Oncotype DX assay. And we looked at women across multiple different sites, all of whom had Oncotype DX test done, and found that when you looked at patients who had a low Oncotype DX risk score, we actually found that the image classifier within the low Oncotype could actually substratify a subset of patients who actually had far worse outcomes. Now, again, these are all women that, according to the Oncotype DX categorization, should have avoided chemotherapy or did not benefit from chemotherapy. And yet we found, of course, it's a limited study, but we still found in our limited study that about 12% of women who had a low Oncotype DX risk score, in hindsight, might have actually benefited from adjuvant chemotherapy because they had more aggressive behavior. On the other hand, when we looked at the high Oncotype DX, so all these women who had been identified as candidates for adjuvant chemotherapy, we found that something like 57% of those women had a low risk score based on the image classifier, suggesting, again, suggesting in quotes, that in hindsight, these women potentially could have possibly avoided adjuvant chemotherapy. Now, of course, I will happily grant that to really validate this, you need to think about validating it in the context of an RCT. And one of the things that we're trying to work with Kevin on is looking at the RxPonder trial, which is a randomized clinical trial, to really validate these algorithms. But again, it shows the ability of these image classifiers or these explainable AI algorithms in the context of the current standard of care to add additional value and additional granularity. And so this is my tongue in cheek, so hopefully you'll indulge me. So why should we care about algorithms like this, right? And one is the cost, like I mentioned, in places like India, you can't afford a $4,000 test, and so being able to develop a test that costs pennies on the dollars can really have an impact in LMICs. The rapidity of the test is another consideration. You can do this analysis off the cloud and get the result back to the oncologist really in a matter of minutes. You're not destroying any tissue, you're digitizing the tissue. And therefore, you can have an impact on a global basis. In other words, you have an inexpensive, fast, reliable, accessible prognostic breast cancer test that's priceless. For everything else, there's MasterCard. All right, but jokes apart, one of the things, like I emphasized, is the importance of validation. And this is data that we reported at the San Antonio meeting a couple of years ago. We're working with SWOG, the Southwestern Oncology Group, in particular, Alistair Thompson and colleagues. We were able to analyze images from the SWOG S8814 clinical trial, Kathy Albain study, and showed in a blinded manner. So we basically ran the analysis on the slides, sent the results back to the biostatisticians at SWOG, and they did the analysis and showed that there was a strong association with survival in this completed clinical trial. But one of the things that we also have been really deeply thinking about is how do we ensure that these algorithms are truly equitable? How can we ensure that these algorithms work across different populations? I'm sure a number of you are aware of this study that came out in JAMA Oncology in 2021, showing that for a test, this Oncotype DX test that's been around for over 20 years, data first came out showing that it actually was not prognostic in Black women. And so one of the things that we have to be very mindful of as we're developing these AI algorithms is to ensure that they work across different populations. And one of the things that we have been doing very intentionally in our group is working with multiple partners across the globe. In fact, right now, courtesy two different NCI-funded grants, a UO1 as well as an RO1, we're working with partners at the Tata Cancer Center in Mumbai in India to essentially get slides of breast cancer patients in India and applying these algorithms to analyzing patterns in South Asian women. And so one of the things that we've been doing and finding is that when you look across different populations, there are subtle differences in the stroma, there are subtle differences in the collagen architecture across different populations. And some of our early data is suggesting that there may be a need to really develop more population-tailored models that allow for more accurate risk stratification and outcome prediction in certain populations. And so this is data we presented at San Antonio a few years ago showing that creating a population-tailored model in South Asian women actually resulted in better outcome prediction compared to a population-agnostic model. Well, it's not just about invasive disease. This is a paper that's under review, work we did with Sunil Badwe and Mangesh Thorat in the UK, and looked at DCIS. And with DCIS, of course, the big question, apart from the prognostic question, which of these DCIS will progress on to invasive ductal carcinoma, there's also a predictive question. Which of these women will benefit from radiotherapy over and above surgery? And of course, again, we don't have a very good answer to that question. And so what we were able to do in this approach, this was a blinded study using samples from the UK ANZ trial, which was a 2x2 trial of DCIS in the UK. We got the images of the trial and we ran our image-based risk algorithms to look at immune density, essentially use the machine learning to go in, identify the individual tumor infiltrating lymphocytes on H&E images. And we're able to come up with a risk score based off the TIL count alone. And we were able to show not only were we able to predict risk of progression to invasive ductal carcinoma, but also that there was a predictive aspect to the algorithm, specifically showing that in patients with a high TIL score, based on the AI, based on the machine learning, that those were the patients who were going to receive maximal benefit from radiation therapy. So not only were we able to demonstrate a prognostic value, but also specifically predictive value, predictive benefit. This idea of TILs is something that we've been very, very interested in. And apart from just looking at the number of TILs, we've also come to appreciate that looking at the spatial interplay and the spatial architecture of TILs is really important. This is a study that we presented at last year's AACR meeting, work we're doing with Priyanka Sharma in Kansas, but now are extending to working with Sunil and Bill Lee and Kevin here at Winship, where we're looking at the spatial interplay between tumor-infiltrating lymphocytes and cancer cells on standard H&E images of triple-negative breast cancer. We were able to demonstrate in a multi-site manner that we could predict outcome and survival in these triple-negative breast cancer patients. And in this case, we actually had data both from Kansas, but also from the National Cancer Center in Lima in Peru, demonstrating that these features are not susceptible to variations from individual labs and that they really are prognostic of outcome across multiple different sites across 2 different continents. Apart from the work we're doing on pathology, we're also doing a lot of work in radiology. This is a publication from a couple of years ago in CCR, where looking at the tumor-associated vasculature on MRI scans of breast cancer patients undergoing neoadjuvant anthracycline therapy, we showed that looking at the twistedness of the tumor-associated vasculature, we were able to find an association with pathologic complete response. Again, it wasn't the amount of tumor-associated vasculature, it was the twistedness, the convolutedness of the tumor-associated vasculature. And what was fascinating in the study was we found that those patients who tended to have a smoother vasculature tended to be more strongly associated with pathologic complete response. On the other hand, those patients that tended to have a much more twisted, convoluted vasculature tended to be associated with a lack of response in the neoadjuvant setting. The other piece we've also been very interested in is looking at metastatic breast cancer. This was a publication that came out last year. This was actually showcased on the Winship bulletin. And we actually showed in this study that we'd done earlier with the Cleveland Clinic and University Hospitals that looking at metastatic HER2-positive breast cancer and specifically looking at metastatic lesions in the liver, by applying our radiomic-based analysis, where we're looking at subtle textural and heterogeneity patterns of these metastatic lesions in the liver, that we were able to predict in advance of treatment as to which patients were going to respond to CDK4/6 inhibitors. And this was all based on routine CAT scans, right? So this is another important consideration that what we're talking about here are fairly standard routine radiologic scans. And we showed that we were able to predict accurately response as well as survival in these patients from the Cleveland Clinic. All right, moving on to lung cancer. This is an area that we've been doing a lot of work over the last 10 years. Similar to the work I showed with breast cancer, the focus really has been on predictive assays. This is a paper from about 3 years ago where we looked at the question of adjuvant chemotherapy. Now, of course, one can argue that in the age of immunotherapy, is this really relevant? But certainly at the time that we did this work, it certainly was relevant. And what we were able to show, again, was that the spatial interplay between the immune cells and the cancer cells on standard H&E images was not only prognostic of outcome in the early stage setting, but specifically was predictive for added benefit of adjuvant chemotherapy in the early stage setting. I mean, I won't belabor all the details of the study, but this was a fairly comprehensive study that we did. The only limitation, the real limitation was, unfortunately, in this particular study, we didn't have a clinical trial to validate against. So these were all institutionally treated patients. And so probably some potential bias there. But across the board, we found that when you looked at patients who had received surgery alone, that we were able to identify those patients who would receive the added benefit of adjuvant chemotherapy. And conversely, looking at patients who got surgery and adjuvant chemo, that the image classifier was able to identify patients with truly low risk, suggesting that in hindsight, they could have potentially avoided the adjuvant chemotherapy. We followed this up with another study that was published a couple of years ago